All right, hi everybody, this is Marianne. Welcome to another Riot. Today we're very excited to have Mitul and Ndaba talking about fast-track research. Before we start, just a reminder of the exciting program we have this year up to July. And if you see on the 11th of June, we have an amazing conference that is planned. We have great speakers coming and an amazing program, and I really want you to sign up for it and spread the word to other people from other universities, other colleagues. Anyone who really is interested in open science is welcome. So please spread the word. It should be an amazing day, and hopefully we will all enjoy it together. Now, today we are in lockdown, all of us, because of COVID-19, and a lot of research has been done towards this. And it made research, it's research that has been done in a different way than we usually do. It's fast-track research. We have to act very fast to respond to the pandemic, and that's what the talk is about today. And we will have two amazing speakers. Mitul, who is currently the head of Neuropharmacology of the Neuropharmacology group in the Neuroimaging department and director of the Center of Innovative Brain Therapeutics, and the deputy lead of the NIHR-BRC for mental health. And Ndaba, who is a clinical research fellow in clinical neuroscience. And his studies include trial design and management, and especially in pharmacological and neuroimaging studies. He is also a repurposing lead on the Center for Innovative Therapeutics. These guys are amazing as friends and researchers, and I'm very excited to listen to their talk. We're going to start with Ndaba, and hopefully Mitul will join us in a tiny bit because he's been delayed. Because guess what? There's an R&D meeting that just had to happen right now on this study that they're building up. Yes, because science moves that fast. So without further ado, Ndaba, would you please share your screen, and then we'll hear you talk. Thank you.
Great. So hopefully everyone can hear me now. So I'm just going to try and share my screen. We did do a practice session for this, so this should be a lot easier than it seems at this point. Can everyone see that yet, or not yet?
Not yet. You're sharing the screen for the Microsoft Team at the moment.
Yeah, okay, let me try that once more.
That's good, cool. Oh, no, go back to the presentation. You have to stay on it.
Okay, hang on. So what can you see at the moment?
So I see you're sharing your Teams live event thing.
Yeah. Okay, so that's not sharing anymore, right?
No.
Okay, let me try and bring up the presentation.
So once you have the presentation on, don't touch anything and just start presenting. Don't go back to Teams.
Okay. You're not seeing it yet, though, are you?
No.
Okay. Okay, can you see that now?
Not yet.
Ah, okay. So I'm going to go to desktop.
Ah, hang on. Something is appearing. Yes, cool. It's your present. Wait.
Okay, perfect. So you can see that now?
So I think what needs to happen is I need to send this, like, oh yeah, perfect. Okay, that's working.
Yeah. Okay, apologies everyone. They did go through with this with me the other day, but clearly I wasn't paying as much attention as I thought. But welcome everyone, and thanks very much, Marianne, for the introduction. And as Marianne said, this is very much an evolving experience. So this is fast track research, our experience, and the experience is actually happening as we speak, because Professor Mitul Mehta is currently in a meeting with R&D, as explained, discussing things related to approvals for the study. So this is very much hot off the press, or very much still in the press. So we'll keep you posted. So my background, as explained, is clinical, but obviously the last few years I've been doing clinical research, and a lot of that recently has circled around the area of drug repurposing. And so that's really the context of what this fast-track research is about. Drug repurposing is not a new phenomenon. This is something that has been around for a very long time. And in fact, it's not even a niche phenomenon. This is something that happens pretty much in every therapeutic area of medicine. And it's something that is actually so common that people often won't even acknowledge it as an obvious, different, or even innovative approach. But what is changing is that drug repurposing is becoming more established and more popular for various reasons that I'll go into in a second. But just to take a brief step back to actually look at what the term means. So drug repurposing is sometimes known as drug repositioning, or reprofiling, or indication expansion, etc. And so what it involves is establishing new medical uses or indications for drugs that are already known, including approved or discontinued shelved experimental drugs, essentially. So taking a bit of a step back again from that to look at where drug repurposing fits in. So this is a slide that I'm sure many of you are familiar with. This is essentially how we look at the drug development process. So going from what happens when someone has a really good idea of what drug could work in a particular setting, and then moving that forward through preclinical work, of course, in its various guises, and then from phase 1 all the way through to reviews, FDA or MHRA, and mass production and prescribing in practice. And of course, as most of you I'm sure will understand, this is not necessarily a linear process. So you've got, neatly here, phase 1, 2, 3, etc. But as we all know, sometimes these things will move back and forth as people try and figure out ways to just translate it. And at the bottom here, you've got essentially the probability of success. So essentially what that looks at is what are the chances of something going from a great idea that someone has to something that actually makes the market. And of course, there is a massive attrition rate as you go down those various phases. And again, depending on the therapeutic area, but if you look here, oncology, for example, on average, in this particular study in biostatistics, published a couple of years ago now, has a really poor probability of success for various reasons, so down to sort of 3.4%. There's been a lot of talk, of course, about vaccines recently and how quickly those are going to be developed as far as the COVID scenario is concerned. In this particular study, the probability of success certainly seemed better than oncology, for example, but of course the context of that here is that these are vaccines mainly for diseases that have been known about for a while, and of course COVID is a new disease, so that might change the scale a bit. What's different in COVID, of course, is the level of attention and the amount of resources that have been thrown at it in such a short space of time. So that theoretically, anyway, might change the probability of success there. But this slide in general does, I think, underscore how difficult it is to get a drug to market. And so that's something certainly to keep in mind when we discuss this. So the difficulty as far as costs is very apparent, of course, so up to over $1 billion US to produce a drug, but also the temporal difficulty as well. So that just, anything from, sort of 10 to 15 years for a novel agent. And this is where drug repurposing comes in, because again, you can short circuit some of those early challenges, for example, and essentially come further in down the pipeline. And again, this is not necessarily a linear experience here, but certainly that it's possible in ideal situations to shave off anything between sort of 5 to 10 years by virtue of using the drug repositioning format. And of course, that's not only important for patients, getting drugs to patients quickly, particularly with acute or very aggressive therapeutic areas, so various forms of cancer, of course, but certainly in the COVID scenario, in areas like sepsis, for example, it's important to try and have things come to market sooner rather than later for the patient base. But also more widely, economically, obviously, it makes a lot more sense if you can shorten the amount of resources required to get these drugs to market. So this is an example of some very current drug repurposing. Some of these drugs are commonly known. Others have, in the last few weeks, become incredibly sort of infamous, perhaps, in the media. So hydroxychloroquine is being talked about a lot. It was in the news this morning, but famously, of course, it is currently in a study in the White House, N=1, an observational trial as a prophylaxis. So we all await with keen eyes to see how that works out. But actually, in this country, it's part of a randomized trial. So the largest trial that's happening in COVID in the UK. In fact, it's currently the largest COVID trial in the world, with over 10,000 participants that have been recruited, remarkably, over the last few weeks. It's part of the recovery trial, which has several arms. So this is a multiple arm platform run by the Oxford an Oxford group, but actually is a national trial. So it's recruiting over in over 160 actually sites over the country, across the country. So hydroxychloroquine is a drug that actually, in a way, already has some repurposing attached to it. This is a drug that is traditionally part of the anti-malarial group of drugs that's been moved mostly into treating things like rheumatoid arthritis and SLE. Azithromycin is a commonly used antibiotic, which is also on this trial. Interferon beta is a treatment for relapsing MS, and lopinavir, ritonavir are also drugs that are used in this trial, and these are drugs that are previously or still currently actually used in HIV treatment. So this is all very kind of current at the moment. So the trial that we are talking about today is also, of course, an example of drug repurposing, and a drug that actually is very familiar to just about everyone, I would imagine. And interestingly, this drug had its own initial issues with media reporting and infamy, if you like. And this was all kind of picked off by a statement actually that was made by a European Minister of Health in France, I believe, who mentioned use of simple analgesics in the community, and there was discussion around paracetamol and ibuprofen, and that was sort of picked up on and reported. And there was some commentary, including a commentary in the BMJ around this as well, which kind of seeded some confusion around the safety of ibuprofen. And so that was kind of an unfortunate experience, but this was actually all kind of turned around because it was clear how sort of dangerous this could be if it wasn't clarified. So both the European Medicines Agency, the WHO, and within the UK Commission on Human Medicines, an expert working group there, all came out with a statement to say that actually there was certainly no evidence to suggest that there was any properties in standard ibuprofen that could cause issues with either susceptibility to contracting COVID or actually contributing to worsening of symptoms once someone had it. And actually, this has sort of come up, and we'll discuss this a little bit further on, as part of the hesitancy about adopting this trial. But again, the fine print around this is that this drug is actually not standard ibuprofen. This is a lipid formulation of ibuprofen, which actually has quite different properties in the way that it is biodistributed, which has implications for its safety as well as its efficacy. But actually what we are looking at here with this particular drug is acute hypoxemia in respiratory failure, sometimes referred to as ARDS, the criteria around that, which is something that the SARS-CoV-2 virus seems to kind of affect through mechanisms I'll describe. But this is a multi-center, open-label, randomized controlled trial, and it's a sort of later phase trial, again as a kind of repurposing agent, and it's been sponsored by Guy's and St Thomas's and King's College London. So it's essentially looking at the reduction of severity of disease. So this comes under the umbrella of therapeutic treatment in COVID, as opposed to vaccine or a prophylactic treatment analysis. So in a way, the underlying condition that the virus causes in this situation is actually well established, and the inflammation around that is also quite well established. And again, interestingly, ibuprofen has been investigated in that as early as the 1980s, preclinically in multiple murine and porcine models of animals with ARDS, both as a single agent and in combination. And in those studies, again, overwhelmingly, there was an improvement of survival, which was dose dependent. And the mechanism seemed to be this reduction of inflammation, preventing leakage and extravascular changes with lung water, but also other conditions as well related to that we're seeing a lot of in the COVID response, such as hypertensive and pulmonary hypertensive changes and cardiovascular changes as well. So ibuprofen seemed to kind of mediate responses in these areas which improve the survival. And actually this was also validated by some case reports in humans which seem to back some of this. So some recent activity around this is that the group that's been exploring the lipid ibuprofen version of ibuprofen, a drug which is called Flarin, looked at this specifically again in the ARDS model. And if you can see this graph here, so this preclinical study shows very clearly that actually without intervention, the survival in this particular group is quite poor. So about 20% survival here. With standard ibuprofen, that survival goes up to about 40%. But with the lipid version of the ibuprofen, the Flarin, that survival goes up to 80%. So there's a very dramatic kind of change in what happens when you invoke the Flarin response here. And this is to do with something that's sometimes referred to as a cytokine storm that creates this really sort of quickly escalating inflammatory buildup in the lungs, which then impairs the gas exchange. So the co-primary objectives of the study here are to look at reducing disease progression. And again, this is quite acute. So actually, you can see this in a matter of days. And there's again been some case reports suggesting that it could be even quicker than that. So looking at hours. And of course, a lot of this is to prevent people getting to the critical care status. So prevent people getting admission to intensive care, and obviously getting to a point where they need ventilation, essentially. And this has obviously become a big factor in terms of people needing ventilation. And the sample size here actually is modest compared to the other trials I've mentioned. The recovery trials, needing to recruit over 10,000 patients for a signal there. But our study has been calculated statistically to require 230 patients, and we are doing an interim analysis at 142 with a data monitoring safety assessment at 20 patients. We are examining other factors here as well, which include overall survival and reduction in length of hospital stay as well, of course, which are important for services to be able to cope with the pandemic. So as explained before, obviously my background and Mitul's is actually in neuropharmacology and neuroimaging. So that begs the question, of course, how did we end up getting involved in a respiratory infectious disease trial, essentially? So the context for that is this center, so the Center for Innovative Therapeutics, CFIT, which is a center that's sort of going through a kind of rebranding experience to try and reposition ourselves. It's an example, I suppose, in a way of repurposing, as a kind of triage hub where, in a sense, we act as the vehicle for the IOPPN, which is the institute under which we sit, to help accelerate collaboration with industry, and also to look at how that collaboration is designed for the mutual benefit of both academia and the industry. And that's something that, again, is not new, but trying to kind of drive that in a different way. It's not new for academia, obviously, to engage with industry, but a lot of how that works normally, as I'm sure most of you will probably know, is that involves dealing with quite large pharmaceutical companies, for example, in our particular sphere. So some of that is to try and drive creativity at the ground end, innovation at the ground end, to try and engage early with people, to co-design studies, to co-design the pathways for how we get drugs from the bench to the bedside, essentially. I think Mitul's joined in, so
yeah, I'm here.
I don't know if you're around. Hi, Mitul. Welcome. So are you happy to kind of take things forward from this point?
Yeah. I don't know. I mean, you might need to listen carefully to what I say and move the slides forward.
Absolutely. So let me know. So this is the slide I'm on here. So you can start with that, or I can skip to the next slide if you like.
It's from the government data. I mean, some people will be very familiar with this, I'm sure, showing the daily deaths reported. So you can see that, at the moment, where we are in mid to late May, the incidence of deaths is actually falling fairly linearly at the moment. You can see the weekends on that graph, by the way. So that's why the rolling averages are more important than daily numbers. If you go to the next slide, Ndaba, this is a timeline, really, of how we got started on this trial and where we are now and all the things that have happened in between. So I apologize in advance for giving you a fairly detailed view of our lives over the last couple of months, but I think it's quite instructive to see what a compressed start looks like for a study. So you can just click away, Ndaba, as I speak. So, initially we had a very good contact with a company called Seek, who are a small pharmaceutical organization. They deal with repurposing treatments. Ndaba leads our repurposing efforts and had a good working relationship with them, which we've expanded. We also work in diagnostics as well. And they contacted the health secretary because they had some great data in mice showing that one of their drugs may be able to reduce the acute respiratory distress that you get in severe COVID-19 disease. They didn't get a reply. Now they only waited a day. But what's interesting is the next day, the next person they contacted was us. And that's not to brag, but I think it's just to tell you that we built up such a strong relationship that they decided that contacting us would be more fruitful than contacting even respiratory physicians cold as well. So who you know, relationships do really matter in getting these things going. We got in touch straight away with the head of KHP, who was able to advise on a CI, a chief investigator, who's been fantastic. So we met with them, we met with the BRC and the R&D leads, we informed them what we planned, we appointed some people, we reviewed all the data, we had various meetings from other people that were thinking about funding it. And then it took a couple of weeks, but we got a consensus on the study design, and then we started the actual awful process of getting studies going. We got added to a database. There was a call for funding. We had a draft protocol produced, and we filled in a lot of forms. Then it got really busy. We were advised that we had to go for national prioritization. The scheme had been brought in to help align research across different hospitals and across different cities. But we also got approval for fast track ethics and MHRA, where they were able to review it within a couple of days. They're amazing, really. We engaged with the clinical trials unit, statisticians, finalized the protocol. We got the funding. We heard about the funding, and a week later we got the funding. We did actually write the proposal. And this is at a time when Ndaba and I sat at a desk next to each other and wrote the proposal, which seems really strange now. We had a kick-off meeting arranged by Guys, which they arranged a fast-track approval and kick-off meeting, and we were the first study to go through it, and that was fantastic. And then we had to prepare all the submission packs for the approvals. We had to wait a little bit while for the internal approvals to send them off. We sent off our NIHR prioritization request. The drug arrived, and we got the paperwork in within a week. Started preparing the logistics. The next one, please, Ndaba. Then it all started to go a bit wrong. We weren't badged by the NIHR, and all that meant was that we weren't given priority status to go national. Now we're a local study, so we weren't interested in going national, and the system had a clause which allowed local studies to proceed in order to maintain diversity of experimental approaches. But the R&D had interpreted that as an unapproval, and we weren't to proceed, and they stopped the study. And then it started to get interesting. So we were convinced that they'd misinterpreted. So we engaged with the head of the health schools, who went to the government, who then actually wrote when they wrote to the R&D committees. That letter arrived the next week, but Robert Lechler wrote to the R&D committees, and at the same time, KCH disengaged from the study for the same reasons. We lost our chief investigator that we had at that time. We went through a few chief investigators because they were linked to the NIHR anyway, and then Robert Lechler really tried to get the R&D committees back. So that worked. The study was unpaused. All our replies went back to the ethics. We had other sites starting to get interested, and then the final one, probably, I think, Ndaba. And then our approvals were in, and then something weird happened, which is the NIHR sent a letter confirming that we weren't badged. And despite what had happened with the CMO letters and Robert Lechler, the head of the health schools, they paused our study again. And we felt that this intransigence was going to be the death of us. But, they gave other reasons, including the fact that numbers were falling. And so we had to re-engage with them, which we've done, and so we're back up and running again. So hopefully we'll be having results soon. So I'll hand over to you, Ndaba, for this.
Yes, thanks.
Tour of what we've been up to. So, one thing I want to add is that, you know, that process that we just showed you, which is over a couple of months, that normally could take a year. And that bit in the middle with compressed activity over 2 weeks normally would take a minimum of 2 months. Yeah.
Great, thanks, Mitul. Yeah, so as you can see, I mean, there's a lot of activity there, and also a lot of sort of confusion all round, really, or difficulty in understanding, anyway, of how some of these processes work, which, you know, may not be surprising given the fact that this is very much an evolving, dynamic situation. And part of the difficulty, I think, obviously, for people who are involved in this whole area is estimating the resources required for implementing these new layers that you need, again, as the crisis evolves itself. And as most of you will probably know, normally the MHRA and REC approvals, for example, will be adequate, and the sponsorship from the institution or the groups that are running the trial. But in this case, at some point we had all of that, and that didn't seem to be adequate, at least was not interpreted to be adequate in some quarters. And so the addition of the badging, which obviously was done to meet other challenges well, also created this other level of difficulty about what was required and what was not required. And of course, the last bit of that is that actually, in order for these processes to work, you need buy-in from the local R&D committees, who obviously are the gatekeepers of what's possible within a particular institution, you know, the capacity and the capability. And if there's differing interpretation about what these directives mean, or what these guidance missives might mean, then obviously that creates difficulties for the whole fast track process. So we thought we would kind of try to unpack this a little bit and look at how research generally works, and what the models might be in a crisis situation. And with different crises, I'm sure each of these models have applied at some point. So normally there's business as usual, which obviously is a description of how local centers will initiate a study and get the approvals that we've alluded to and carry on, essentially. But in a crisis situation, there actually may be benefits to trying to create a large consortium model, which actually has benefits in terms of numbers. As I mentioned before, the recovery trial, for example, is to date the largest COVID trial in the world. I think over 10,000 people in that particular project. So you are unifying things, and you can do that because essentially you get all the institutions or hospitals or trusts around the country to all essentially buy into joining that project. But of course, the downside to that may be that you have a much smaller number of projects, and that actually you can't invoke the level of creativity that you want. And that's sort of related to this next one, where you have a centralized top-down effect. So with the consortium model, you can actually do that locally if you have people that engage with each other. So you can have the sort of bottom-up model where researchers from institutions around the world around the country can organize themselves in order to do that. The approach that seems to have been done more comprehensively with this particular crisis is more of a sort of top-down one, where things have been centralized in order to move quickly in that way, and resources deployed centrally. And an alternative to those is to have a larger number of project platforms, and then kind of have those, you know, operating with much smaller numbers in each, but actually you have more creativity perhaps, or more diversity in the approach that you have as a result of that. Feel free, Mitul, to jump in with any comments.
Actually, I will add one comment to that, Ndaba, is that the centralised fast track scheme came in, and it's, you know, well considered, you know, in order to get people working together. But the issue that arose out of that was that we had a very small number of projects, and of course there's a high risk if the drugs don't work. And what's happened in the UK is that after the centralised fast track scheme was running for a short while, they actually brought in another scheme of project platforms with smaller numbers in each. So obviously there is a recognition that there was a loss of diversity, at least just by looking at what has been done. Yeah.
So finally, you know, to kind of look at how all of this works, or certainly our experience within that, we thought we'd kind of analyze it to a few issues here that have sort of come up. So we've kind of discussed the institutional frameworks, you know, of how you can deploy this, and they have, you know, positives and negatives in each one, you know, and I think, you know, our experience actually has comprised both of the sort of positive and negatives of some of that, you know. So it's been a difficult experience, but actually there have been some really kind of positive things that have come out of this in terms of the support, and in terms of the broad nature of the collaboration, you know. And of course, you know, that speaks to the kind of robustness of the institution. If you can have people that work in a neuroimaging department collaborating with people in respiratory and ICU medicine, and being able to kind of deploy links to other departments and other agencies to get things done. So so that's been important as well to kind of recognize. But of course, in a rapidly moving system, there can be misalignments in how people interpret things. So the messaging, I think, is quite important. So for example, the messaging all the way down to R&D groups, as well as researchers, as well as government agencies, of course, about what the parameters of those frameworks are. So not only including what you want to be involved in a particular thing, but also being clear about what's either being included or not excluded from those groups, if that makes sense. So I think part of the difficulty with this is that the metrics as to how people conduct their work can differ. And so obviously everyone is trying to do all of this to try and get therapeutics to patients. But also, you know, we understand that we live in a framework where actually in order to do that, you need funding as well for that. So in some cases, some people's roles are more geared to organizing that funding stream, for example, or creating an environment that is conducive to bringing more funding in. And other people's roles are more geared to actually sort of directly trying to get things to patients more quickly. So there can be sort of differing approaches which are related to roles. And I think a comparison here may be how researchers, so I'm sure a lot of you will be familiar with this particular challenge, is the challenge of grant funding. So, you know, most researchers would acknowledge that actually their primary role theoretically is to, you know, produce scientific output, but actually quite often a co-primary role or a competing role for sure is actually grant funding. So those two things are supposed to go along in parallel, of course, but sometimes, particularly in acute crisis situations, can become slightly competing issues or issues that can trip each other up, if you like. And so, so for example, one of the things that was slightly unclear to us in terms of where we stood was, on the one hand, we had funding, which, as I said, we had lots of support from various places like KHP, IOPPN, BRC, for example, which were all kind of underlying the support that we had from these various places, from that side of things. But also there was difficulties about what that meant in terms of badging from another point of view, and some of those things perhaps may have been cleared up earlier on in terms of all of us sort of understanding the details of that a bit more, or being clear about what was being said. So again, Mitul, I don't know if you have anything further to add with these final thoughts.
No, but I think we've got a couple more slides. We want to end on another note, actually, if that's okay with you, Ndaba.
Yeah, just absolutely.
So I'm going to talk about the Great British Wellbeing Survey and the Cogent study, which are both live at the moment. And one of the interesting outcomes of this is that we're reading a lot more about the symptoms that you might get with COVID-19 disease, and it's well known, actually. So could have known this before, that the hospitalization that's required, particularly the ICU admission, and maybe even the ventilation, leads to long-term cognitive impairment. So by having a look at this literature, talking to these clinicians, and being exposed to the research a bit more, these things catch your eye, because I work in cognitive neuropsychology and psychopharmacology, so this became very interesting. So what's amazing is that in some studies, every single patient receiving mechanical ventilation showed impairment. And what's not on the slide is that in follow-up studies, one particular study, which is a 5-year follow-up, showed that 1 in 5 patients, 20%, continued to have cognitive impairment. So it became clear to us that even though the numbers were going down, and obviously people were unfortunately dying, but people were surviving as well, more people were surviving, that we have potentially an ensuing problem coming up with large numbers of people being impaired at some level and maybe not being able to function in their life in the same way as before. So the model on the right, the picture on the right, shows you the current ideas about what might be leading to the cognitive impairment. It is not well understood. It could be some of the experiences in ICU, sedation, delirium, ventilation, hemodynamic instability. It could be because of other physiological processes ongoing, could be related to dementia risk, blood-brain barrier dysfunction, for example, reduced oxygen, pre-existing brain injury. What we do know is that those factors together might have individual contributions, but we know that overall these patients have a high risk of not doing well in the long term. So we put together a team, the names are here. We've created online questionnaire, series of tests. We've linked up with Adam Hampshire and Pete Hellier, who did the Great British Intelligence Test, and we have put this online at the moment. So the next slide, if you don't mind. The hypothesis being that obviously they'll be impaired, those with severe disease. So in the foreground, what you've got is at the top, we've got previous studies looking at cognitive impairment. They are all pretty much cross-sectional. You only really know about these patients in ICU when they're in ICU. We're in a situation now where you've got a population, and some of those population members are going to end up in ICU. So pre-pandemic, we had 27,000 data sets of people who agreed to be recontacted. And also, I can't tell you precise numbers, but we added significantly to that in January, which is arguably all pretty much pre-pandemic. We had a very small number of cases in January, fairly isolated as well. All agreed to be retested. They will be re-invited very shortly, and that will allow us to have a long-term impact of the severe disease. We've also got We've got a grant that's being discussed at the moment. Hopefully we'll get funding. We have collaborators who will do follow-ups in subgroups to look at workplace function, occupational effects. Maybe if we get the funding, we will bring people in for imaging, long-term effects on other markers such as inflammation, for example. So there's a lot of potential outcomes here we can understand. The tests and the questionnaires are at the bottom. The background of this slide was prepared by Adam Hampshire. Thank you, Adam. And what he did was he added in a couple of free text boxes into the online survey and tests, where he asked people to put down a few words that they were negative associations of the current lockdown and positive associations, and he just put together a word cloud after we got 80,000 people in as well. So you can see we can start to get interesting thoughts developed quite early on, and by going online, of course, we can get rapid and large data sets together. So it's interesting to see how we can come in from one angle, which is trying to treat the respiratory problems, and we're coming out with a trial where hopefully we'll show a positive drug effect. We might not, that's the point of the trial, but we're also coming out with other questions around our usual area of expertise around long-term cognitive impairment and everyday functionality in these people. So thank you very much. Happy to take questions. I hope my mic.
Thank you very much. This was very inspiring, and it shows the roller coaster you've been through in the last 2 months, which is quite crazy. So we do have a few questions, and the first one is from Olivia. She asks, do you think that timelines for research approvals will change in the future as a result of COVID-19?
No. That would be nice, wouldn't it? I'll tell you what, though, we know all the people really well now, so we don't, and that does seem to make a difference, unfortunately. I wish that were the case, that it will be true, but without saying too much, I can imagine there are some people who like to hold on to their decision-making power. I would, yeah, but I think if we had another crisis, which of course we don't want, I think we could deal with it better. And I think the idea that we do have fast-track systems, and they can work, and they can have unintended consequences, shows that we can be responsive. Yeah.
Yeah.
Sorry.
So I was just going to quickly add, I think I agree with Mitul. I mean, I think what this does, though, is it actually does throw into sharp focus where some of the bottlenecks might be. I think when you have a situation like this, it becomes very clear what the sticking points are because of the sense of urgency around it. So I think if people want to improve the timelines, you know, the decision makers on this, this is a good opportunity to do that.
Yeah,
again,
there were definitely people who bent over backwards to rethink their processes or you know, accelerate their processes, and there were certainly people who dug their heels in and insisted on their process mapping as it was. And there might be obviously a good reason for each individual process, but that was quite right. We know exactly where delays are more likely to occur.
I see. So I do have a follow-up question on that, because I mean we have seen it in our professional lives, how long it takes for a study to start, especially a drug trial. I can think of one especially, but is there a reason why it's so slow then? Does it make the decision making better? And then is there a risk then for fast track research to be, you know, dangerous in that sense that we don't have the same evaluation of the research around it? And does it lead to then noise around everything that is related to COVID-19?
Sure. I think it's important to say we have all the same approvals you'd normally have. We have all the same meetings and all the same people asking us useful and useless questions all the way through. So, you know, in that respect, you know, the intensity of activity has gone up, but the activity hasn't gone down. We're in a situation where there was a diktat to stop or pause all non-COVID research. So that opened the pathway to allow the fast tracking. I think in a normal situation, you've got competing studies going on, and that does affect the timelines as well. And that's kind of unavoidable. That's a capacity issue. But there are stages where I think we can accelerate the process, certainly, and we've learned that as well through the previous experiences before the pandemic.
I see. And so I have another question quickly before I move on to Sam, who has questions as well. But do you think that because we are in this pandemic, the incentive and the benefits are bigger and seen as bigger than the risk, and then things are being accepted more or faster because it seems to be a good solution for the pandemic we're in a sense that if the ethics committee is made of lay people and expert people, maybe people who are in the center of this, who have people in their families who are sick and everything, want to get rid of this and find a solution, something that helps, will be biased and accepting this faster because we are all, you know, in the middle of it.
Well, I mean, maybe you're right. I don't know about all the studies. In our experience, that has not happened.
Okay.
Yeah.
Brilliant. Oh, we have a couple of other questions. Sam, would you mind asking your questions first, and then I'll ask the other ones?
Sure.
No, well, I don't mind. I give the audience an opportunity. Do you want to just do that? I can defer to them.
Sure. So we have a. Sorry.
Oh, yeah. Now we have questions about ibuprofen safety. Can you see the questions in Ndaba? You can answer that.
Hang on, I need to stop sharing my screen, don't I?
You distract people with the word cloud.
I know, exactly. All right.
Yeah, so from Claudia and Chris, we have two questions.
Can you see those, Ndaba, or we can read them out to you if you like?
Let me, I think I'm in now. 1 second. Actually, it might just be easier if you read them out.
Okay, so Claudia asks, could you please comment on the information that was spread in the news that ibuprofen should not be taken when experiencing COVID symptoms because, in quotes, it could make them worse, and people should instead stick to paracetamol?
Yes, so it's a good question. So this was something that was that actually has caused us quite a bit of grief through this whole process. So there were some reports very early on, I think sometime in February, from a French health minister. Yeah, exactly. So who kind of made this comment about taking ibuprofen and its potential danger. That was linked to some, again, unsupported detail about ACE2 receptors, which are seen to be receptors that allow for the virus to sort of take hold and enter the body, about, you know, whether ibuprofen had more of a sort of link to those particular receptors. But there's no sort of peer review kind of detail to back that up. And so actually, again, this was pushed back on very firmly by a number of different agencies, including the EMA, the WHO, and even in the UK, you know, the UK Commission on Human Medicines was very clear about two things. The first is that there's certainly no evidence to suggest that actually taking ibuprofen increases susceptibility to getting the virus. And secondly, that ibuprofen would worsen the symptoms of someone on this. And actually, beyond all of that, again, as I sort of mentioned previously, there's been a lot of both preclinical work in murine porcine models dating back as early as the 1980s, but also in human studies. There's in fact, a randomized trial using intravenous ibuprofen in ICU patients, which showed no danger at all. And this is a group that's even more sort of severely ill than the population we would be tackling. That showed no adverse events, but actually seemed to show a lot of promise for efficacy along the line. So the data is very much actually, you know, the other side of that whole sort of argument in terms of where this would be. Now, I mean, something that we were quite sort of careful about and cautious about, this was not an invitation really for people to be taking ibuprofen in the community, because the doses that we're talking about are different to doses that people would normally be taking in the community. And obviously in a trial like this, you want to have that kind of expert clinical monitoring of these patients. But at the moment, there's really nothing to say that ibuprofen makes anything worse in this situation. But certainly in the context of our trial, the doses that we are talking about and the way that we control the trial, of course, that happens in a hospital setting as opposed to in the community. So this is not an invitation for people to be taking ibuprofen. And the last thing I'll say about that, of course, again, is that we're dealing not with standard ibuprofen. So we're dealing with a lipid-based formulation of the drug, which again is different in terms of its safety factors, its biodistribution, but also from the point of view of the trial, we feel there are differences potentially as far as the efficacy at comparable doses. So hopefully that answers your question.
Yeah.
Great, thank you. We have another question from Chris. He asked, what avenues are you considering for fast-tracked publication or dissemination, and how do you ensure rigorous peer review included?
Well, I can answer that. I mean, obviously, we're in the midst of actually setting up and about to start running this trial, so our attention hasn't been focused on publication, dissemination as much. But you know, we've got two issues here. The first is that, you know, we do have a protocol. In fact, the protocol's publicly available. All the outcomes are all published so that anyone can see that. So we do have a situation where we know the outcomes and we can publish them on clinicaltrials.gov or whatever. And that might lead into alterations of decision making prior to any peer-reviewed publication, of course. That's true of any trial. It's not unique to us, and that's a situation that already exists. In terms of publication, I think we would expect the journals to be open to the same sort of peer review process as they have been. But looking at the publication record so far, there are a lot of papers coming out very quickly. So I would share the concerns and share the question about what level of rigorous peer review is ongoing here. Because the data coming out is really important. I mean, we use the data that is published to power our study. You know, we don't have much choice but to use the data that's published to power our study because it's a rapidly evolving situation. So I think we have to work, you know, in partnership with the journals to ensure we get proper peer review.
Brilliant. We have two more questions. In fast-tracked research, are there different mechanisms to evaluate and monitor safety and efficacy closely? No, there are not different mechanisms. It's exactly the same mechanisms. If anything, they're probably applied more in our study. They're applied more frequently. So if anything, there is a higher level of monitoring in some of these fast-track studies because obviously people are very, very interested in the outcomes. There is a risk, and we've seen it in some of the studies coming out of the States, of early evaluation of results, which might be too early and lead to rapid decision making, which has a risk of blocking further research, which we've seen with remdesivir and the FDA. You know, arguably they've approved it too early. That only has the effect of blocking research. Why would you take part in a placebo-controlled trial of an approved drug when there's nothing else? So there are some risks involved, but no, I don't think the monitoring is less. If anything, it's more. Brilliant. Last question before Sam's. Bartek is asking, is anyone else in the world doing similar study with ibuprofen?
Nope.
No, currently no. You know, there's maybe one other group which is sort of doing a kind of observational thing with ibuprofen. In Denmark, exactly. But that's more a sort of monitoring of kind of retrospective monitoring of outcomes in people who might have happened to be on it, as opposed to a prospective randomized trial.
I mean, I could add to that. I mean, you know, I daren't say too much because things are changing very rapidly, and we're doing a trial because this might not work. You know, it's very important that we do the trial. And if it doesn't work, so be it. But if it does work, you know, we are talking about a treatment which is oral, can be taken in the day, and costs 25p roughly per capsule. So we are next week talking to researchers in lower and middle income countries about whether they would want to adopt the trial if we get early indications of safety, of course, which we'd want before we ask someone else to look at running the trial. So yeah, we would run it elsewhere in the world if it does happen in partnership with other people.
Brilliant. I think that's it for the Q&A. Sam, do you have questions? I think you did.
Yeah, I do have a question. Bartek did ask a follow-up one, though, I've just spotted and published. But I just asked mine very quickly, because it's sort of jumping off the back of the question about disseminating your work and requiring rigorous peer review. Sure. I don't know whether either of you want to pass comment on the sort of the recent crisis of confidence in, say, bioRxiv and other preprint servers that have been sort of swamped with preprints researching COVID-19. It seemed to have shaken a lot of people's trust in that as a quick way to kind of almost get post-publication peer review. But I wonder whether any of you have any comments on that, or you can pass on that and I can move on to another question.
Yeah, I mean, you know, this is something that came up when it was first launched, that people will be just putting anything out there. And, you know, that seems to be a low-level concern, really. But this crisis has really brought that into focus again. I don't really have any thoughts because I haven't had time to reflect on it. But I do think it does raise some issues about the fact that effectively you can self-publish this work, and bioRxiv gives it a veneer of credibility that it might not deserve. And that is certainly something to reflect upon. I wouldn't begin to question the existence of bioRxiv. I think it's a very good thing, but I think it does make us reflect.
Okay, and just last couple of questions. In regards to sort of sharing data, and whether you intend to share your data and allow others to see the raw data. Yeah. So yeah, is that true? Are you going to do that?
I have no idea. I mean, okay. Our intention is always to share our data from all our studies, and we do have permission on our ethics to share the data. So yeah, I don't see a problem with that. Although, to be honest, we haven't really thought about that beyond making sure it's in the ethics, which I think is where we are at the moment.
Cool. And the other thing that has cropped up in the coronavirus is sort of rapid registered reports. And I guess, I mean, are you familiar with this? And what are your thoughts on sort of rapid review registered reports? If you don't know, I can quickly.
Yeah, I mean, you might need to explain it a bit. The only thing I would say, without knowing all the details, is that we probably don't have time. So.
Yeah, I see. So would you say it's more amenable to other projects that aren't sort of randomized clinical trials of your scale?
Yeah, I think capacity is an issue that we have to consider there. Yeah.
Right. Okay. I don't know whether Ndaba, you have anything to say on that. If not, I'll just go on to Bartek's question, which was if applying for this or other C19 project fast track routine again, what would you do or not do to have it processed even faster?
Yeah. So I mean, I do have something to say about that. I think. What certainly would help is at the very beginning of the process, identifying, you know, what all the different levels are, you know, that we talked about in terms of, including things like badging, for example, and making sure that everyone, from the researchers to the R&D teams to anyone else that might be involved in the process, understands exactly what those different levels mean, and which are required and which are not required for the study to run. So I think that would have certainly saved a lot of time. So in our example, for example, you know, we assumed that things like having sponsorship might kind of also be much more closely aligned with capacity, capability type issues locally, and that would not need to be revisited again in certain ways. And so it's kind of clarifying those sorts of things and making sure that everyone understands what bits mean and what things might hold the study up, and who the sort of key decision makers are in the process, and identifying them early.
But I must say that, you know, it's very difficult to think how we do it again, because some of the blocks we've experienced and some of the turgidity, let's say, were from processes which were developed and introduced after we started developing the study as well. So there were things that were difficult to anticipate. Obviously, being linked in to decision makers at a high level, would probably have made it easier early on. And we did try to do that as well, and we got some support from some people, like, you know, the Vice Dean for Research and the head of the health schools. And maybe naively, we thought that having support from seniority meant that we would get a smoother ride through, and that's not necessarily true. Yeah, but interesting.
It is definitely interesting.
Yeah, yeah. We know who holds the real power now. Anyway, we shouldn't say too much. It's being recorded.
Exactly. I think we're going to come to an end. Thank you very much, Yeti. That was a very, very brilliant talk, very insightful. Next week we have Daniel Lakins at 12. And enjoy your day, everybody. Thank you.
Thanks very much. Bye.